I first took David's plant physiology class when I was a sophomore at Reed, and it was one of the first upper division biology classes that I took, and it really changed my perspective about what I wanted to study. I became fascinated with plants and how they work and all the amazing things that they can do. And the summer after I took that class, David was kind enough to offer me a position in his lab to work here, and that's how I got started on the ascorbic peroxidase project, doing the genetic work that eventually gave rise to these plants.
The project came about as a result of some long-term interest I've had in plant antioxidants, especially vitamin C. And the story with vitamin C and other antioxidants is that plants use that to protect themselves against oxidative damage, which is especially likely to happen in times of environmental stress. So for several years we've been looking at different stresses and trying to demonstrate how important antioxidants like vitamin C are for plants.
I'm able to examine how they tolerate stress differently from regular wild-type plants. So I am responsible for producing the drought treatments and then monitoring how they respond to this drought treatment and all other stress treatments as well. So like herbicides, bright light, flooding, and things like that.
We actually have two different genes we've identified, one for the biosynthesis of vitamin C and one other antioxidant gene that's involved in scavenging of hydrogen peroxide. So we have these two separate genes. They've been cloned in the laboratory and manipulated and then reintroduced back into Poplar in such a way that they will be overexpressed or overactive. We expect that those plants will be tolerant of more stress than a regular wild-type plant.
Immediate applications would be that poplar itself is grown as a crop to produce paper and biofuels, so having increased stress tolerance in poplar would in itself be a huge advantage, but also it would demonstrate that this same metabolic engineering could be a success in other crop plants, especially food crops. And that's been a huge goal of agriculture and plant physiology for hundreds of years, is to increase stress tolerance. So there's definitely the potential for wide applications of this project.
As is often the case, Reed students pretty much take ownership of a project. I mean, the good ones do, like Lauren, and they're looking ahead to solving problems. And just a real pleasure to work with. Lauren's actually doing the vast majority of the hands-on bench work and the greenhouse work, and she's done her homework in terms of developing the methods and knowing which questions to ask. So it's pretty much my job just to point her in the right direction, turn her loose.
It's been great because working with David has felt more like a collaboration than a typical professor-student relationship. We've really been able to share our ideas about it and plan out the steps of the experiment together.